Dogma, Dogma, Dogma. Coming soon to a biology lab near you. How does the information contained in your genetic code get into proteins that work and function around your body?
Transcription.
Transcription is the process of mRNA being made using DNA. This happens in a cell nucleus and is done by a protein called RNA polymerase. This protein unzips the DNA and uses it as a template to make the mRNA. Yes, zipper, but not the zipper made by this guy in 1914. A zipper made of
Hydrogen bonds.
The DNA is split into two strands, the template strand, or the antisense strand, and the coding strand, or the sense strand. The template strand is used by the RNA polymerase as a template to make the mRNA, and so the new piece of mRNA will end up matching the code on the coding strand.
But what direction does this happen in?
Glad you asked. That happens in the 5' to 3' direction using the energy from
RNA nucleoside triphosphates,
which form new phosphodiester bonds.
Okay. But how does the RNA polymerase know which nucleotides to put down? And in what order?
The RNA polymerase puts down new nucleotides using complementary base pairing. A goes to U, T goes to A, C goes to G, and G goes back to C.
transcription.
At the HL level, there's a couple more things to talk about. It all starts with
Initiation.
To initiate, the RNA polymerase binds to a section of the DNA on the gene called the promoter region. Each gene has its own promoter, and each promoter contains DNA sequences that help the RNA polymerase and the helper proteins attach to that section of DNA. In eukaryotes, helper proteins called
Transcription factors.
bind to the promoter first, helping the RNA polymerase attach to the DNA. Some of these transcription factors increase gene expression and some decrease it, but we'll get to that later. In eukaryotes, most initiation begins at something called a Tada box. It's a sequence of genetic code that contains lots of A's and T's, which makes it much easier to pull the strands of DNA apart, because they're connected via 2 H bonds rather than the 3 between C and G.
Elongation.
Elongation is the process I described earlier, where RNA polymerase moves from the 5' to 3' direction, unzips, produces the complementary base strand according to complementary base pairing, and uses the energy from RNA nucleoside triphosphates.
Termination.
Transcription finishes at a DNA sequence that is called the terminator sequence. It's a sequence of nucleotides that marks where the mRNA should end. This signal is then recognized by an enzyme that cuts the RNA transcript and releases it from the RNA polymerase.
Well, if that's how it gets from the DNA to mRNA, how does it get from mRNA to protein?
Translation.
Translation speed round. The 4 bases of mRNA, A, U, C, and G, need to be converted into the 20 different amino acids that make up polypeptides. This is done through codons, a sequence of 3 bases that represent one of the 20 amino acids, or a stop codon. For example, AAA, or AUG, or UAG. Each codon of 3 base pairs in the mRNA codes for 1 amino acid within the polypeptide chain. However, there are multiple possible codons for each amino acid. In addition to coding for specific amino acids, certain codons signal the start of a gene, and certain codons signal the stop of a gene.
But how is this process done?
The process of translation is done in the ribosomes. The ribosomes are made of protein and ribosomal RNA, or rRNA. Each ribosome is made up of a large and a small subunit. The small subunit of the ribosome contains an mRNA binding site, whereas the large subunit contains tRNA binding. The tRNA binding sites contain the A site, the P site, and the E site, or exit site. Transfer RNA, or tRNA, has 4 key regions. The acceptor region, which carries an amino acid. The anticodon, which connects with the mRNA codon via complementary base pairing. It also has a T-arm, which connects with the ribosome via the E, P, and A binding sites, and a D-arm, which connects with with an enzyme that activates the tRNA.
Initiation. Again?
The small subunit on the ribosome first binds to the mRNA. It moves along, similarly to transcription, in the 5' to 3' direction along the mRNA until it reaches the start codon. The anticodon of the tRNA molecule, as one of the arms, then binds to the mRNA start codon, AUG, using complementary base pairing rules. The large ribosome subunit then binds to the small subunit.
Elongation. Again?
Yes, this process is continued down the mRNA as the ribosome moves along it via separate molecules of tRNA. Each tRNA aligns with codons according to the complementary base pairing, and each tRNA molecule will carry a specific amino acid and form peptide bonds to make the polypeptide. The ribosome will continue moving along, synthesizing the polypeptide chain until it reaches the stop codon, and then the mRNA and polypeptide chain, or protein, is then released.
Genetic code.
The genetic code is the process by which, or the rules behind which, translation happens. It describes which orders of base pairs or base nucleotides in the mRNA turn into which amino acids, and this is codons. There are several aspects of the genetic code that are important to understand. Number 1, it's universal, which means every single organism uses the same code. It all evolved only one time. It's unambiguous, which means no codon specifies for more than one amino acid, so AAU would only mean one amino acid, could not make multiple. It is also degenerate, which means that several different codons specify for the same amino acid, and it's redundant, which means that many codons are degenerate unnecessarily. So, you'd only need one codon per amino acid, but we have more.